The evolution of mammalian auditory ossicles is one of the most well-documented and important evolutionary events, demonstrating both numerous transitional forms as well as an excellent example of exaptation, the repurposing of existing structures during evolution. In reptiles, the eardrum is connected to the inner ear via a single bone, the columella, while the upper and lower jaws contain several bones not found in mammals. Over the course of the evolution of mammals, one lower and one upper jawbone lost their purpose in the jaw joint and were put to new use in the middle ear, connecting to the stapes and forming a chain of three bones which transmit sounds more efficiently and allow more acute hearing. In mammals, these three bones are known as the Malleus, Incus, and Stapes. The evidence that the Malleus and Incus are homologous to the reptilian articular and quadrata was originally embryological, and since this discovery an abundance of transitional fossils has both supported the conclusion and given a detailed history of the transition. The evolution of the stapes was an earlier and distinct event. Riker Corp theory. Following on the ideas of Etienne Geoffroy St. Hilaire, the relationship between the reptilian jawbones and mammalian middle ear bones was first established on the basis of embryology and comparative anatomy by Riker and advanced by Corp, and this is known as the Riker-Corp theory. In the course of the development of the embryo, the incus and malleus arise from the same first pharyngeal arch as the mandible and maxilla, and are served by mandibular and maxillary division of the trigeminal nerve. The discovery that the mammalian Malleus and Incus were actually homologues of visceral elements of the reptilian jaw articulation ranks as one of the milestones in the history of comparative biology. It is one of the triumphs of the long series of researches on the extinct theromorph reptiles begun by Owen, and continued by Seeley, Broom, and Watson, to have revealed the intermediate steps by which the change may have occurred from an inner quadrata to an outer squamosal articulation. Yet the transition between the reptilian jaw and the mammalian middle ear was not bridged in the fossil record until the 1950s with the elaboration of such fossils as the now famous Morganucodon. There are also more recent studies in the genetic basis for the development of the ossicles from the embryonic arch, and relating this to evolutionary history. BAPX1, also known as NKX 3.2, is the vertebrate homologue of the Drosophila gene bagpipe. Others are DLX genes, PRX genes, and WNT genes. Definitive mammalian middle ear. The mammalian middle ear contains three tiny bones known as the ossicles, Malleus, Incus, and Stapes. The ossicles are a complex system of levers whose functions include reducing the amplitude of the vibrations, increasing the mechanical force of vibrations, and thus improving the efficient transmission of sound energy from the eardrum to the inner ear structures. The ossicles act as the mechanical analog of an electrical transformer, matching the mechanical impedance of vibrations in air to vibrations in the liquid of the cochlea. The net effect of this impedance matching is to greatly increase the overall sensitivity and upper frequency limits of mammalian hearing, as compared to reptilian hearing. The details of these structures and their effects vary noticeably between different mammal species, even when the species are as closely related as humans and chimpanzees. Evolutionary history. Definition of mammal. Living mammal species can be identified by the presence in females of mammary glands which produce milk. Other features are required when classifying fossils, since mammary glands and other soft tissue features are not visible in fossils. Paleontologists therefore use a distinguishing feature that is shared by all living mammals, but is not present in any of the early Triassic therapsids. Mammals use two bones for hearing that all other amniotes use for eating. The earliest amniotes had a jaw joint composed of the articular and the quadrata. All non-mammalian amniotes use this system, including lizards, crocodilians, dinosaurs, and therapsids, so the only ossicle in their middle ears is the stapes. but mammals have a different jaw joint, composed only of the dentary and the squamosal. In mammals, the quadrata and articular bones have evolved into the incus and malleus bones in the middle ear. Summary of the fossil evidence. Here is a very simplified family tree of the various lineages involved. Tetrapods, plus, amphibians, a reptiliomorphs, amniotes, plus, sauropsides, synapsids, pelycosaurs**, therapsids, mammals. The first fully terrestrial vertebrates were amniotes. Their eggs had internal membranes which allowed the developing embryo to breathe but kept water in. This allowed amniotes to lay eggs on dry land, while amphibians generally need to lay their eggs in water. The first amniotes apparently arose in the late Carboniferous from the ancestral reptiliomorphs. Within a few million years, two important amniota lineages became distinct. Mammals' synapsid ancestors and the sauropsides, from which lizards, snakes, crocodilians, dinosaurs and birds are descended. The earliest known fossils of all these groups date from about 320 to 315 m years ago. Unfortunately, it is difficult to be sure about when each of them evolved, since vertebrate fossils from the late Carboniferous are very rare, and therefore the actual first occurrences of each of these types of animal might have been considerably earlier. The pattern in most of the following sections is that each successive, more advanced group started with the more primitive jaws and ears of its predecessors, then developed more mammal-like jaws and ears, and so on. The evolution of mammalian jaw joints and ears did not proceed neatly in lockstep with the evolution of other mammalian features. In other words, jaw joints and ears do not define any except the last of the various stages into which paleontologists divide the evolution towards the mammalian. Anatomy. Early tetrapod in amniota ears in modern amniotes. The middle ear collects airborne sounds through an eardrum and transmits the vibrations to the inner ear via thin cartilaginous and ossified structures, which usually include the stapes. But the earliest tetrapods, amphibians and amniotes probably did not have eardrums. In fact, eardrums apparently evolved independently 3 to 6 times, in stegocephalians, in anorans, in synapsids, in diapsids, perhaps separately in anapsids. If turtles are not modified diapsids, probably in femur-iomorphs, and possibly in some temnospondyls. In all basal members of the three major clades of amniotes, the stapes bones are relatively massive props that support the brain case, and this function prevents them from being used as part of the hearing system. But there is increasing evidence that synapsids, eureptiles and parareptiles developed eardrums connected to the inner ear by stapes during the Permian. Early therapside jaws and ears. The jaws of early synapsids, including the ancestors of mammals, were similar to those of other tetrapods of the time, with a lower jaw consisting of a tooth-bearing dentary bone and several smaller posterior bones. The jaw joint consisted of the articular bone in the lower jaw and the quadrata in the upper jaw. The early pelycosaurs most probably did not have tympanic membranes, and the massive stapes bones supported the brain case, with the lower ends resting on the quadrates. But their descendants, the therapsids, probably did have tympanic membranes, and these probably were in contact with the quadrata bones, and the stapes bones were still in contact with the quadrates but functioned as auditory ossicles rather than brain case supports, so the therapsides' quadrates had a dual function as part of the jaw joint and as parts of the hearing system. Twin-jointed jaws during the Permian and early Triassic. The dentary of therapsids, including the ancestors of mammals, continually enlarged while other jaw bones were reduced. Eventually, the dentary was able to make contact with the squamosal, a bone in the upper jaw located anterior to the quadrata, allowing two simultaneous jaw joints, an anterior, mammalian, joint between the dentary and squamosal and a posterior, a reptilian, joint between the quadrata and articular. This twin-jointed jaw can be seen in late cynodonts and early mammaliforms. Morganucodon is one of the first discovered and most thoroughly studied of the mammaliforms, since an unusually large number of Morganucodont fossils have been found. A Morganucodon is an almost perfect intermediate in this respect between the higher mammal-like reptiles on the one hand and the typical mammals on the other. Mammal-like jaws and ears. As the dentary continued to enlarge during the Triassic, the older quadrata articular joint fell out of use. Some of the bones were lost, but the quadrata, the articular and the angular became free-floating and associated with the stapes. This occurred at least twice in the mammaliforms. The multituberculates, which lived from about 160 m years ago to about 35 m years ago, had jaw joints that consisted of only the dentary and squamosal bones, and the quadrata and articular bones were part of the middle ear, but other features of their teeth, jaws and skulls are significantly different from those of mammals. In the lineage most closely related to mammals, the jaws of Hadrocodium suggest that it or a very close ancestor may have been the first to have a nearly fully mammalian middle ear. It lacks the trough at the rear of the lower jaw, over which the eardrum stretched in therapsids and earlier mammaliforms, and the absence of this trough, which suggests that Hadrocodium's ear was part of the cranium, as it is in mammals, and hence that the former articular and quadrata had migrated to the middle ear and become the Malleus and Incus, but Hadrocodium's dentary has a bay at the rear which mammals lack, a hint that its dentary bone retained the same shape that it would have had if the articular and quadrata had remained part of the jaw joint. It has been suggested that a relatively large trough in the jawbone of the early Cretaceous monotreme Taenolophos provides evidence of a pre-mammalian jaw joint, because therapsids and many mammaliforms had such troughs, in which the articular and angular bones docked, and therefore that Taenolophos had a pre-mammalian middle ear, and therefore that the mammalian middle ear ossicles evolved independently in monotremes and in other mammals. But a more recent analysis of Taenolophos concluded that the animal was a full-fledged platypus and the trough was a channel for the large number of nerves that collect signals from the electrical and vibration sensors in the bill, and therefore that the trough is not evidence that Taenolophos had a pre-mammalian jaw joint and a pre-mammalian middle ear. Ironically, Rich and Vickers-Rich were among the authors of the 2005 paper on which they later cast doubt. A recently discovered intermediate form is the primitive mammal Yanoconodon, from 125 million years ago in the Mesozoic, in which the ossicles have separated from the jaw and serve the hearing function in the middle ear, yet maintain a slender connection to the jaw via the ossified Meckel's cartilage, which in more advanced mammals dissolves during development. How these changes affected hearing, the frequency range and sensitivity of the ear is dependent upon the shape and arrangement of the middle ear bones. In early synapsids such as the pelycosaurs, the quadrata and articular had to function as the jaw joint, and this severely limited how far these bones could be modified to alter the frequency range of the ear. But once these bones were no longer involved in the jaw joint, variations which affected hearing would not also affect jaw joint function, and this allowed unconstrained evolution of the mammalian hearing apparatus. By the Jurassic, the typical mammalian ear had evolved, in which the angular had become the tympanic annular, while the articular and quadrata had become the Malleus and Incus, respectively, connected in series with the stapes. This series of three bones acts as an impedance matching system to improve sound transmission and allow enhanced hearing. The transition between these two states is one of the most well documented and supported in all of evolution, and newly discovered fossils from this transitional period have recently improved our understanding of this transition. But they also suggest that it was not a simple linear process from the early therapside jaw and middle ear to the modern mammalian anatomy. Natural selection. It has been suggested that natural selection could be a factor in the preservation of the structure of the middle ear in mammals. Many of the earliest mammals were quite small, and the dentition indicates that they were insectivorous. If they were warm-blooded, like modern mammals, then they could have been nocturnal. This fits with the popular image of small, nocturnal insectivorous mammals surviving in niches not accessible to the large, dominant contemporary dinosaurs. The enhanced hearing, particularly in the higher frequencies, would be helpful for nocturnal animals, in particular for detecting insects. This scenario is consistent with selective advantage being a contributory factor to the transition. Summary: by extrapolating the developmental morphogenesis of genetic studies into the early mammal fossil record, evolution of the middle ear in early mammals provides an integrated case study of how development has impacted, mechanistically, the transformation of a major structural complex in evolution. Zhe developmental patterns in Mesozoic evolution of mammal ears. While the stapes is present in many types of tetrapods, the addition of the Incus and Malleus in the middle ear is a signature feature of mammals, distinguishing them from reptiles and all other vertebrates. They therefore have the appearance of representing a discontinuity in the tree of life. But in the early 19th century, it was hypothesized that these bones are not a total novelty, but are the equivalents of two bones which non-mammals have in their jaws. This hypothesis made sense, not only of the existence of these middle ear bones, but also of certain other features of the anatomy, such as the paths taken by nerves in the head. As evolutionary biology began to be expanded upon, this relationship became treated as one of common descent. For the evolutionary explanation to make sense, it seemed to demand that there would be a transition in function between being part of the feeding mechanism in the joint of the jaw and serving only in hearing, and this would mean that somehow there had to be an intermediate connecting these two quite different functions. With the discovery of Morganeucodon and other fossils, there were concrete examples of this. There was a double jaw joint, the older reptilian, as well as the newer mammalian, in the same animal. This meant a confirmation of the pattern of inference from comparative anatomy to evolutionary biology. The earliest mammals were generally small animals, probably nocturnal insectivores. This suggests a plausible evolutionary mechanism driving the change, for with these small bones in the middle ear, a mammal has extended its range of hearing for higher-pitched sounds which would improve the detection of insects in the dark. Natural selection would account for the success of this feature. There is still one more connection with another part of biology. Genetics suggests a mechanism for this transition, the kind of major change of function seen elsewhere in the world of life being studied by evolutionary developmental biology.